Yeah, I
Now,
can see a
can you
screen.
see this? Excellent. I'll try to go on full presentation mode and you can tell me whether it's displaying okay.
Yeah, I can see it.
Yeah, great. So yes, thanks a lot again, Abby, for the introduction and thanks for the invitation. So what I'd like to tell you a little bit about today is some of the current research in the development of biomaterials for tissue engineering and regenerative medicine, and in particular how it's possible to engineer these biomaterials to control stem cell phenotype, so the behavior of stem cells, and how this has implications for these fields for biomedical applications. So this is all work that was carried out at Queen Mary University in London. And to give you a brief introduction first to the field of regenerative medicine, which is a particular strong topic of research in this university. This is a field that is about um, developing solutions to help uh, cure, address diseases and conditions in human health. And biomaterials can come at various different levels in these fields that can be from biosensing, so the detection of molecules that might be associated with the development of conditions. If we want to um, detect these molecules at early enough stages to be able to diagnose patients as early as possible. Materials are extremely important in order to interface between life science and the molecules we want to detect and the interfaces that are going to give a readout. It can be also for the development of antibacterial coatings, so that these coatings and materials will prevent bacterial adhesion and will prevent infections, for example, during surgery and implantation of devices and materials. So preventing these events is really important. Also, drug delivery applications. So there, the materials will allow the controlled release of therapeutics so that a patient will benefit from low dosage over a prolonged period of time at clinically relevant doses. So biomaterials can be important in all of these applications, as well as in a particular field that is known as tissue engineering. And there, the aim and the focus is really to try to regenerate the tissue. So rather than introducing a material that is going to just plug a hole or bring two tissues, for example, two bones that have been fractured together, not necessarily helping the correct repair of the tissue. Biomaterials can be designed to orchestrate a regeneration process that will give rise to the formation of a new healthy tissue, functional and with the correct structure. And inherently, the engineering in the field of biomaterial is about realizing that the materials we use that are typically synthetic, so they are man-made, need to be interfaced with biological system, and they do not do that normally. Cells have developed mechanisms to sense their environment. They have cell membrane receptors that can sense the presence of molecules or other cells in their environment, but they have not developed the ability to sense and respond to these synthetic materials. So a lot of the design of biomaterials is about bringing strategies to confer to these materials the ability to interact specifically with cells and the tissues they're going to be interfacing with. And in particular, we're going to be interested in regulating the behavior of stem cells because they hold really huge potential in the field of regenerative medicine. Stem cells are cells that can differentiate into different cell lineages so they can um, change their behavior or phenotype, we say, to adopt different behaviors. For example, and I'm going to try to I'm going to try to display a pointer. No, I can't see it. I don't know if you can see my cursor. Maybe not.
We can see a cursor, yeah.
Oh, you can see my cursor. Okay, so that's great. A stem cell will be able to differentiate and adopt a different phenotype or behavior and turn into different types of cells. For example, muscle, blood, bone, skin, and so on. And this obviously has a huge implication in regenerative medicine because it means we are going to be able to tap in this capacity of some cells to regenerate normally a tissue that might be diseased or damaged during trauma. So this has a lot of application in the field of tissue engineering. Um, for the production of biotherapeutics, turns out that many stem cells secrete many molecules that are beneficial for promoting repair. And in the development of the next generation of 3D in vitro model for mimicking diseases and testing therapeutics, organoids and organ on chip, and I'll touch on this topic in my lecture. So the key aspect to realize with stem cell and their biology is that although all of the cells that constitute an organism and our body share the same genetic material, the same genome, the same genetic code, they have completely different behavior. Cells within our brain behave fundamentally differently from muscle cell or bone cells. And this is despite the fact that they all share the same genetic code. So how do we get from this unique genetic code to cells that display completely different behavior? And this is about development from one single stem cells that is uh, able to differentiate into all of the cells of our organism. We're going to have a gradual change in behavior to generate the various lineages that will form our body and form this organism. And that's obviously in contrast to the genomic diversity that we can see across species and between member of one single species. Obviously, there is also the diversity, much less, but there is still some diversity. So development is about going from one stem cell sharing one genetic code to a multiple of cell lineages that have exactly the same genetic code but completely different behavior and able to create the different tissues of this organism. For example, a first case is that of the epidermis. So that is the upper layer of the skin, and that's what protects us from our environment. We see here at the top the layers of dead cells that are eventually going to shed and that are protective. And right underneath, we have a number of other layers of differentiated cells that are originating from this single layer of stem cells that are at the boundary between the dermis underneath and this epidermis. And they adhere to this very thin layer of protein called the basement membrane that define the stem cell niche. This is something that we'll hear and we'll discuss further in the next few slides. Stem cells like to reside in a very defined environment that is known as the stem cell niche. That's an environment that is going to give them signals to display the stem cell phenotype and um, uh, maintain the normal structure and function of the tissue. So this is the case in the interfollicular epidermis. but also in many other tissues. Another example in the small intestine, we've got again a layer of epithelial cells that are facing the gut and the intestine, and that are there to absorb nutrients and transfer them to the blood supply and microvasculature underneath within the villi, within these villi. And at the bottom of these structures, we see a very particular region in which stem cells will reside. And they have a role again to replenish all of the cells that are forming this epithelium and that are responsible for the normal function of the intestine and absorption of nutrients and so on. So really important again to have this very special unique location to regulate the role of these cells. If these cells somehow disappear, gradually the organ will stop functioning properly and fail. So it's extremely important to maintain this structure, but tapping into this potential has also huge impact in the field of regenerative medicine and for the repair of the particular tissue or organ of interest. We can also define a number of important concepts in the field of stem cell biology. Stem cells in particular need to display potency. So they need to display a capacity to differentiate into a different lineage or different type. They will adopt a different phenotype, so display different properties. And some stem cells will be able to differentiate into any cell lineage. These are called totipotent stem cells. This is the case for embryonic stem cells that at early stages in the embryo can then give rise to all of the cell lineages that are present in a single organism. Other cells are known as multipotent stem cells, and these will only be able to differentiate into a few of these lineages. This is the case of mesenchymal stem cells, maybe you've heard of, that can differentiate into lineages such as bone, fat, or neural lineages. And finally, we've got unipotent stem cells that only have the capacity to differentiate into one lineage, often that will undergo what we call terminal differentiation. So they differentiate into this lineage and eventually will die. And this is the case of the interfollicular epidermis and keratinocytes, or intestinal stem cells in the crypt in the gut, as we've seen in the previous slide. Now, another important property of stem cell is that they need to be able to divide infrequently and retain the stem cell properties, this capacity to eventually differentiate. So in other words, as they divide, we need to have two cells that are generated, one of which needs to retain this stemness, this information that is key to then be able to regenerate the lineages that are potentially disappearing during normal tissue homeostasis and function. And the other cell, the other daughter cell, will be able to first divide many times. This is often what is known as the transit amplifying stage before differentiating into the cell lineage it is supposed to. Underlying this process, there are two different possibilities to orchestrate such asymmetry in the division of the stem cells. The first is known as divisional asymmetry. So the division itself gives rise to two cells that are inherently different, one of which retains stemness and can go back to the stem cell stage. And the other one is able then to differentiate. More often, it is environmental asymmetry that is found in many tissues. Stem cells will divide, and then there is asymmetry in their environment that will give rise to different fate of these cells. So one retaining stemness and its capacity, whereas the other will start undergoing differentiation process and changing phenotype, changing its properties. And this environment that will determine whether stem cells retain their capacity or change phenotype is really key. It will drive this asymmetry, and it's a collection of different signals. These signals can be the presence of soluble molecules that will bind cell membrane receptors, growth factors, or cytokines, for example. It can be physical signals through the extracellular matrix, so the scaffolds that cells are adhering to and that are giving us structure and mechanical properties. And cells are able to, through again receptors, bind to some of these molecules and sense the physical properties of these materials, their mechanics, their topography, and so on. We'll see a little bit more of that later. Or the binding of cell-cell membrane receptors and interactions with neighboring cells. This is another important signal that can regulate cell phenotype. All of these signals together will impact on transcription, so the decoding of the genetic material. Single, again, genetic material shared by all cells within an organism, but that's this context that changes how this code is going to be read and eventually translated into protein expression. And that eventually will give rise to a change in cell behavior, cell properties, whether it is in terms of differentiation, migration, for example, during wound healing, or death, because controlling cell death is extremely important, and this is seen often as a key step that is not well orchestrated in diseases such as cancer. So in the field of tissue engineering, the game is really to harness this potential, taking stem cells and their capacity to regenerate tissues and combining them with biomaterials that are going to guide this behavior and implantation protocols. And together, this will allow to address and treat diseases, potentially regenerating tissues following trauma or injury. And there are many mechanisms via which stem cells have been proposed to impact on tissue regeneration. For example, they could contribute to the replenishment of cells that have been lost during disease or during trauma, and that this cell mass needs to be replaced. So the stem cells that are introduced may be able to differentiate into these cells, or they may stimulate by the secretion of factors the cells from the neighboring tissues to migrate in and replenish these cells that have been that are missing now. They might also stimulate cells to other cell types to come into the tissue and contribute to the repair function. Importantly, vascularization of the regenerating tissue. In order to sustain the regeneration of the tissue, there needs to be a new network of blood capillaries that has to be generated in order to supply the tissue with oxygen and nutrients. So in many cases, this vascularization is thought to be really key to help the regeneration of the tissue. And so in my lab, we're interested in controlling this microenvironment that is really important to regulate stem cell behavior using biomaterials. And we do that at multiple length scale, from the nano to micro scale, where we're going to be interested in controlling, in particular, cell adhesions. So how cells adhere to the extracellular matrix and to the materials that we're using for regenerative medicine, to the micro to tens of micro scale, where we're now going to be interested in controlling cell shape and polarity, so the orientation that cells may take and may adopt. In vitro, this is an information that is lost. We'll see later on. and it's possible to recreate using microfabrication platforms. And as we go towards the millimeter scale, increasingly we're going to be interested in controlling the microenvironment and the geometry of this microenvironment to regulate the formation of multicellular clusters. So aggregates of multiple cells from which we will start seeing the emergence of tissue-like structures that will recreate some of the structure and functions of normal tissues. So I'll give some of these examples as well at the end of my talk. Um, and so an important property of the microenvironment that was recently identified to control stem cell phenotype, in addition to the biochemistry, so the type of proteins and ligands that may be binding to cell membrane receptors, is the mechanics of the matrix and the environment. And this was found to impact on a very wide range of behaviors, from cell spreading, how much adhesion and how much spreading cells are able to generate with a substrate, to their differentiation. So again, their ability to alter their behavior, to adopt different behaviors, or the retention itself of stemness. And in particular for stem cell transplantation, it is extremely important to be able to expand, to allow the stem cells to proliferate in vitro before reimplanting them. Well, the mechanics of the materials that are used for the expansion, for the proliferation of these stem cells in vitro, will have an impact on their ability to retain stem cell properties. And depending on the mode of transplantation, these materials are going to be extremely important. In its simplest form, stem cells can be transplanted by simply injecting suspension of stem cells, and this can be very successful in some cases. For example, in bone marrow transplant, stem cells can be directly injected in the bone marrow. And because there is confinement within the marrow, this is something that can be relatively efficacious. So the stem cells are going to be retained in this environment and will be able to accomplish their function, secreting factors or differentiating and so on. However, in some tissues, this is not something that will be possible. And for example, for the treatment of myocardial infarction, it was proposed that stem cell could contribute to the repair of the cardiac tissue. But how to inject stem cells in this environment where there is a lot of flow, and cells may not be retained at the surface of the tissue? So how do we make sure that they are going to home in the disease tissue and not go somewhere else? So several strategies have been proposed. One was to simply induce the formation of a cell sheet that can be cohesive and that can be formed in vitro before harvestment and transplantation on a target tissue. That may require a materials to allow the adhesion of the cell sheet to the target tissue, but in itself, that just requires the formation of a cell mass that is transplanted. And there are different ways of doing so, but one very successful way was to introduce what we know as a thermoresponsive material at the surface of a tissue culture dish or tissue culture plastic, that makes this surface hydrophobic at body temperature, and that therefore allows proteins to absorb and cells to adhere and form a cell sheet. But that has the property to switch to a hydrophilic behavior when the temperature is lowered down to room temperature, below a transition that is near 32 degrees. And therefore, at room temperature, this material becomes hydrophilic, starts swelling, and releases the extracellular matrix proteins that have been generated by cells, and therefore release the cell sheet. So this can then be transferred and implanted to a tissue of interest. And this has been a strategy that was very successful in many cases, from the treatment of macular degeneration to myocardial infarction, and in particular in the field of skin regeneration, where it's possible now to recreate relatively large sheets of skin and epidermal tissue for their implantation, and saving the life of many patients, for example, suffering for severe burns, but also allowing the treatment of conditions such as epidermolysis bullosa that are fatal. So stem cell technologies have really revolutionized the field in the last few decades. However, in many cases, it's not possible to form cell sheets, or it's not a scenario that is practically translatable into the clinic. So it might not be possible to implant a cell sheet, in particular when it is for treating internal organs. For example, in myocardial infarction, this would require open chest surgery, and surgeons are interested in developing solutions that only require minimally invasive solutions. And therefore, it's interesting to develop solutions in which we're going to inject a suspension of stem cells together with a biomaterial that can flow, so it can be injected, but that will eventually be able to set. So it's going to set at the surface of the tissue and retain the stem cells in place to allow them to deliver, for example, the therapeutics that they will secrete. So the biomaterials that we're going to develop for these applications will require to be flowable, so we can inject them, but to set easily in relatively simple conditions, and eventually also to display and to provide the right set of signal to the stem cells to secrete the therapeutics they need to secrete to promote the regeneration of the target tissue. And this can be engineered through the engineering of the matrix, and in particular often hydrogels that are going to be delivered in this way. This might involve the mechanical properties of the matrix, how stiff or soft this matrix is, the topography, whether we have fibers, whether it's more of a continuous, whether it is porous matrix, and so on. The adhesion that cells can make with this matrix and engage mechanically and physically with this matrix, as well as other factors such as growth factors that may be present in this matrix that will be important in modulating and altering the cell behavior. And an important parameter that has emerged recently is the development of matrix that have controlled degradation, so that cells are initially trapped and locked in the position where we want to implant them, but eventually can locally degrade this environment and start regenerating the tissue. And this is something that we harnessed in a biomaterials design recently that allowed controlled degradation mediated by cells. So cells release the proteins that are going to degrade this matrix locally and in a controlled fashion. And that allows to regulate and control the ability of cells to protrude, to spread within this matrix, rather than completely degrading the matrix relatively quickly. So it's this fine-tuning of the local degradation that seems to be really important. And in turn, we also found that this has an impact on the ability of these cells, so in this case, mesenchymal stem cells, to secrete factors that are important in recruiting other cells from neighboring tissues to orchestrate the normal repair mechanism. Importantly, endothelial cells that are going to form the macrovasculature required to bring oxygen and nutrients to the tissue that is undergoing repair. So factors that will accomplish such role include MMP2, SDF1, IL-10, and VCAM1, and we see are highly expressed in some of these matrices displaying control degradation. And in turn, this has an impact on the ability of endothelial cells to be recruited to the target tissue for orchestrating repair. And here we see them in labeled in green. We've got many more cells, many more endothelial cells that are present in the tissues treated with gel plus the cells compared to other control systems. And we can also show that that way we retain the localization of stem cells very nicely at the surface of the epicardium, so the outer surface of the cardiac tissue. And that the treatment with a combination of stem cells and biomaterials promotes a proper repair, increasing the thickness, the wall thickness of the tissue, reducing the production of abnormal matrix, here labeled in red, and improving cardiac function, here that is quantified in the form of the left ventricular ejection fraction. That's a, um, a way of assessing the mechanical function of the heart, how well the heart is able to beat and eject liquid from its cavity. But beyond the field of tissue engineering, stem cells increasingly have been recognized for their potential and the development of advanced in vitro models. And this is a really a big deal in the field of the pharma industry and the development of therapeutics. There is a big gap in these fields between the early assessment of therapeutics under development that are characterized in vitro in 2D cell culture systems and the clinic. Um, Rapidly, pharmaceutical industry will want to test these materials and these therapeutics in animals, but animals do not necessarily provide accurate prediction of the safety and efficacy of therapeutics when we go to the clinic and patients. Animals are different from human. I don't think it should be a surprise to anybody. And so the development of in vitro 3D model is about bridging this gap and providing an intermediate that is often going to be based on human cells and therefore better mimic human tissue structure and function, and allowing us to better screen and assess the efficacy and safety of therapeutics in vitro before we go to animal, reducing animal testing, but maybe also allowing us to better predict the performance of these therapeutics when they go to the clinic. And biologists have recognized that the environment in which cells are cultured in vitro is completely abnormal. Cells do not look in vitro culture in 2D like what the phenotype and the morphology they adopt in vivo. Very a huge heterogeneity, cell shape looks completely different, and whereas in vivo or in 3D in vitro culture, we've got much better reproducibility of shape, structure, and function. And in order to control this behavior, we're going to need to develop strategies to engineer and microfabricate biomaterials displaying the right signals to regulate cell structure and function. And scientists have realized the importance of microfabrication for a few decades now. Already in the 80s, Fiona Watt and co-workers showed that stem cell shape was able to regulate differentiation and proliferation. So when skin stem cells were allowed to spread, they reduced differentiation and increased proliferation, whereas when they were forced to round up, they were induced to differentiate, and that impacted on their proliferation. And since then, many scientists have showed that This can also impact on a range of other phenotypes or behaviors, from apoptosis or cell death to mitosis, so the orientation of the cell division axis, and many other cell phenotypes that are affected by the geometry of the adhesive landscape in which cells are behaving. And so to regulate this geometry and this microenvironment, we can use microfabrication techniques. For example, borrowing concepts from the microelectronic industry, where photoresists can be used to pattern substrates with thin layer of polymers or materials. And this can be done through a photomask that has clear and dark areas through which the light cannot go through, and therefore, light is only going to be able to irradiate on specific areas in the material. And depending on the type of photoresist, that can either induce a cross-linking that will make the material insoluble, or that can trigger a reaction that will make the material soluble and allow the resist locally to be removed. And so here I've got two examples of such negative and positive photoresist. This material can be cross-linked when photoirradiated, and bonds will form between this molecule and neighboring molecules, so forming a network. Whereas in positive photoresist, as I mentioned, the photoirradiation will induce a change in the chemistry that will make the material soluble. In particular, this in the presence of a base and of an aqueous base will dissolve. So we can then use the resulting resist. So we're at this stage here as a master, as a material that against which we can replicate a soft, flexible material that can be cast and then peeled off from the surface of this master. Typically, this material is a silicone, PDMS, that is liquid initially, that can be poured, adopts the shape of the master, and then can be cured. So by thermal treatment, it will cross-link and result in a soft elastomeric material, so elastic material that can be peeled off. And if we introduce features that form microchannels, that can then be assembled into a microfluidic chip, such as the one that is shown on this picture here. Another strategy for building 3D objects, microfabricated, is 3D printing. And now techniques in particular, such as stereolithography, allow to form features that are getting to the resolution of microfabrication. We can't quite get to the resolution of microlithography, photolithography, as was shown in the previous slide, which is in the range of the micron to even submicron scale. But with stereolithography, it's now possible to generate feature sizes that are near 10 micron or so. So we're really getting there. And the advantage is there that we can really build 3D architecture, whether in the case of photolithography, it's only possible to build relatively simple 3D structure, just a few layers that are assembled together. Using 3D printing, it's now possible to generate much more complex architectures. To get back to photolithography and soft lithography, if we take two microfluidic channels and assemble them together, bonding them on either side of the surface of a porous membrane, it's now possible to generate several compartments. After etching of the membrane in the side compartment, we end up with four separate compartments, two compartments that will define where cell culture can take place on either side of a porous membrane that has pore sizes of just the right size to allow nutrient to go through, but to restrict cell migration. And two side compartments through which we can apply a vacuum, for example, that will exert mechanical forces and deformation on this membrane. And you can see that illustrated on this slide. This was then proposed to allow mimicking the architecture of some of the lung tissue, in particular this interface between an epithelial layer that is exposed to air and allows the capture and transport of oxygen in exchange with CO2, and an underlying endothelial layer that will mimic the microvasculature. And by applying pressure or vacuum to the side channel, it's possible then to deform this thin membrane wall here. So all this is still based on this elastic flexible materials, PDMS or silicone, that will pinch the membrane that is separating these two tissues and therefore stretch the tissue surface. So mimicking in a way the mechanical process that is taking place during breathing. And that was found to be particularly important in many cases, in the case of lung tissue modeling, to capture the response of this tissue to pathogens or to nanomaterials, for example, present in pollution, and the impact that these have on the immune response and immune reaction to their exposure. Another particular phenomenon that is really important to recreate in vitro is the formation of a microvasculature. Many tissues are microvascularized, and in the context of drug discovery, it's really important to mimic this interface between a tissue and the microvasculature in systemic delivery, so drugs that are injected intravenously. This is the mode of exposure through which these therapeutics will find themselves to the tissue that is targeted, or to potential tissues on which the therapeutics may have side effects. And so being able to recreate this microvasculature in vitro and interface tissues of interest with this microvasculature is going to be really important. And again, microfluidic chips allow, for example, the formation of a gel compartment that can be loaded with cells, endothelial cells, so these cells that sustain the formation of microvasculature. And this allows the formation of beautiful microcapillaries that are functional. If we inject a solution containing particles, for example, in one of the neighboring channel, these particles will be able to diffuse through these capillaries as they would in a normal microcapillary in vivo. Similarly, therapeutics or molecules that are injected in these side channels will then be able to diffuse, and we will have barrier function restricting them from crossing the microcapillary, mimicking again what could happen if we were injecting a therapeutic intravenously. And that may also help the study of important processes associated in the development of diseases such as cancer. During cancer development, a particular process known as metastasis can take place, in which cancer cells from the primary tumor site can migrate to a secondary site. And sometimes this takes place through the vascular system. So understanding how this takes place and how cancer cells can migrate through the vascular system can be really important. Very difficult to study in patients. How do you follow this process in a patient? It's really impossible. Even in animals, really, really difficult, and perhaps at the boundary of ethical. But in these types of in vitro models, it's possible. We can generate models of the microvasculature in these microfluidic chips and introduce cancer cells and study how they are able to migrate within these microvasculatures. It's also possible to recreate and interface tissues with these vascular systems. For example, again, in the case of skin equivalents, we can generate a microvasculature and place a skin equivalent of epidermal dermal compartment at the surface of this microvasculature, and characterize the structure, showing that this can promote the development of the epidermis and healthy multi-layer formation. But we can also interface more advanced tissue structures to recapitulate their function and more complex functions. For example, cardiac spheroid or cell masses that can beat spontaneously in vitro. They can be formed by allowing the cells to aggregate together. And we can then take these and introduce them into this microfluidic chip, vascularized again, and we see that they can integrate really nicely. So the cardiac cell mass is perfused and integrated really nicely within this microvascular network. And interestingly, they retain then beating. Maybe on this small video, you can see how the cardiac cell mass can retain the ability to beat and allows some of the perfusion of molecules within this microvascular network. And this is really interesting because now we can inject therapeutics that are used, for example, for the treatment of cancer through the microvasculature and mimic their exposure to the cardiac tissue. And sometimes these therapeutics may have side effects um, and have been found to affect cardiac function, perhaps disrupting the beating of the heart. So being able to investigate, study, characterize such side effects early on in a human context, again, but in vitro without requiring animal testing, is going to be really important for a much broader range of therapeutics and molecules. Okay, so I think I'll stop here. I hope I've convinced you that biomaterials design is really interesting and allows to control a range of different stem cell phenotype and really harness their potential for regenerative medicine, whether it is for the delivery of stem cells for tissue engineering or for the modeling of diseases and tissue function in vitro. And I hope that this was interesting to you all. I would just like to finish by thanking all of the researchers that have developed some of this work in the group, as well as collaborators, and obviously the funders without whom support all this research would not have been possible. So thanks again for your invitation tonight, Abby, and thanks a lot for your attention.
Okay. Thank you very much. That was an incredibly interesting talk. I've learned quite a bit from that. Now we'll have a Q&A session. So students, you can put questions in the chat and I'll go through them one by one. So to start off, I've got a few questions myself. So when stem cells differentiate, what sort of intracellular changes occur when it's differentiating
Yes.
into a specialized cell?
So, so very difficult to give one single answer to that. That will depend a lot on the stem cell and the cell in which it is differentiating. It's often a gradual change that will involve the expression of many proteins. Some of these proteins will be there to kind of try to ensure that once the cell has differentiated, it's retaining this phenotype, this behavior. And some of the proteins are going to be associated with the function of the cells that has just differentiated, the new function of the cell that has just differentiated. For example, in the case of skin and the epidermis, the cells that are differentiating start to express a lot of proteins, structural proteins that will form a network, a dense network of molecule um, conferring structure, mechanical properties to the upper layer of the skin, but also barrier function. So our skin is protecting us from the environment. And so usually you have a set of these properties, these proteins that either help maintaining the new phenotype and proteins that are going to be more functional associated with the new function of the cells that are differentiating.
So what controls which genes are turned on and which genes are turned off?
That's another important and very complex question. So there are uh, there are many mechanisms via which genes can be turned on and off, signaling pathways that will regulate these these genes, and importantly, the expression of transcription factors. So these are proteins that associate with the DNA and that will regulate its transcription into mRNA. I'm simplifying a lot. But so eventually, and maybe you will remember in one of my slides, I had a small schematics that was placing this transcription factor at the center. I don't know if you can see that. So we have all of these signals that come from the cell environment. These are physical signals, these are soluble signals, ligands that can bind to cell membrane receptors, all these are cell-cell interactions. Uh, all of these signals give rise to a series of steps, so molecules that can be phosphorylated, that can be cleaved, and so on, that will lead eventually, in the vast majority of cases, to the regulation of transcription factors, so these proteins that can hook up onto the DNA and modulate the translation of this DNA into RNA. And so once the DNA is translated, we're going to see it's going to open up the ability to translate into then proteins. So triggering the synthesis of proteins that are associated with the function of the particular cell type. Does that make sense?
Yeah. Yeah. With the stem cell sheets that stick onto things, what substances are used for the adhesion? So what would allow it to stick on something?
Often, yeah, it has to be very simple and obviously safe. Often it's a fibrin glue that is used. So fibrin is or fibrinogen is a molecule that is involved in clotting. So when we cut ourselves and blood starts spilling, there's a series of reaction that takes place that leads to the activation of this protein fibrinogen that cross-links into fibrin. So it forms a network again of protein that kind of seals the gap and prevents further loss of blood. And so it's possible to use the same proteins to form a glue that will bind the cell sheet to the target tissue. Problem with this type of approach is that often the glue is relatively weak, so there is always a need to develop new biomaterials that will help promoting better adhesion, but obviously without detriment to the safety of the material.
Yeah. And why does the stiffness of the matrix affect stem cell renewal? Why is that important?
So let me try to find a slide where this could be explained a little bit. Um, not sure I've got a slide here. Actually, maybe we can stick to this slide. So normally, when you have soluble factor, they're just either binding or not to the cell membrane receptors. And so it's the presence and the concentration of these factors that is important to the signal. But in the case of matrix adhesion, these ligands that are going to bind to the cell membrane receptors are linked to a, a matrix that is mechanically um, elastic or that has mechanical properties. So it has mechanical performance. We can pull on it and it's going to deform, but not completely flow. And um, when um, when cells exert forces on these ligands, it pulls molecules that are on the other side of the cell membrane and it deforms them. And as it does so, it activates further recruitment of other proteins that will lead to activation of some of these signaling pathways. So as cells exert forces and sense the mechanics in return of the surrounding matrix. These we call mechanosensing events will be triggered and will lead to the activation of some of the transcription factors we discussed a little bit earlier. Does that make sense?
Yeah. And are there any researchers that have inspired or influenced your work and research?
Yes, certainly. There are many. Certainly, my mentors. So I had the chance, for example, of joining the lab of Fiona Watt, who did these amazing visionary studies in the role of the cell shape, but also the microenvironment on stem cell behavior. I come from a chemical background, so I'm a chemist by training, and I had the opportunity to work at this interface between biology and Fiona Watt's lab and chemistry and biomaterial science in the lab of Wilhelm Haag, who's now in Nijmegen in the Netherlands, but who was in Cambridge at the time. And I think that's been an amazing opportunity to learn from experts in these fields and at this interface. That's something that's really unique.
Yeah. And what's your advice for people hoping to go into scientific research?
Well, I think it's fascinating. Research is really exciting. There's always new discoveries to be made. We're always pushing the boundaries. We are learning so much, but we're also discovering so many more new questions, and I think that's what's really fascinating. So uh, if you have an interest in science and in research, I really encourage you to push these boundaries, and it is you that will shape tomorrow's questions. So I think that's up to you to ask the next questions and to find answers to these questions.
Yeah. Okay. So thank you very much. Um,
Thanks a lot, Abi,
yeah,
for your invitation. It was a pleasure.
yeah, it was a great talk, very engaging, and yeah, thank you everyone for joining, and yeah, see you soon.
Thank you.